Hello, everyone. It's my great pleasure to introduce the next speaker, Professor Chunhe. Chunhe is currently an associate professor at Fudan University and also associated with the Shanghai Center for Mathematical Sciences. He has a background in chemistry and also was trained for his postdoctoral fellowship both in chemistry and mathematics departments, and has been doing some exciting work on gene regulatory network dynamics, as well as plotting the landscape of how different cells make decisions and the tipping points that they come across. Looking forward to your talk, Chunhe. Over to you.
Okay, thank you. Can you hear me?
Yes.
Okay, thank you. I'd like to thank Mohit for the introduction and the kindly invitation. It's my honor to have this opportunity to talk about my work. So my title is Stochastic Analysis and Applications in Gene Networks. So this workshop is on the tipping points of a complex system. So I have to say I'm not really in the field of tipping points or early warning signal, but in one of our recent work, we did find something that might be related with the early warning signal. I hope I can illustrate that point. So first, I want to introduce my research interest. So my research field is computational system biology. I have some directions. The first directions, sorry. Okay. The first direction is we use a data-driven approach to predict protein-protein interaction and construct the biological network. The second direction is we construct the mathematical models, and we develop some stochastic approach to study the stochastic dynamics of gene network. For example, we use the landscape and the transition path approach. Then we apply this approach to some specific biological systems, such as cell cycle, cancer, and so forth. Of course, this modeling will predict, will have some predictions which can be further tested in experiments. So today I will focus on the mathematical modeling and stochastic approach part, and also some applications. So here's the outline. I will first talk about the background of stochastic dynamics of gene networks. Then I will talk about our approach, that is the edge landscape and the dimension reduction approach. And then I will talk about some applications of these approaches in self-fit decision networks. Okay, here is an example for the gene regulatory networks. Actually, it's a cancer network from this classic Cell paper. Here, each node represent genes or proteins, and each link represents some regulations. So in the field of systems biology, there are some questions that people would like to ask. For example, why a gene network so stable to perform functions? Another question is, how do gene networks perform their functions? So there's why and how question. So from the mathematical modeling perspective, we know this network is complex. For example, if the number of genes is n and the number of states for each gene is m, so the total number of possible states in this network will be m to the power of n. This number will increase as m and n increase. So this number will increase exponentially. So we know this network has a huge state space. So there's some relate to the why and how question. There's some issues that we would like to address. For example, how can we calculate the dynamics of this high-dimensional system? Also, how to explore the transition between different states. Specifically, is there any quantitative way to quantify this transition between states? Because from a biology perspective, they will care about the surface transition and how to regulate this surface transition. So this is, of course, a complex problem because gene network is very complicated. So this Waddington development landscape provides some clue for this problem. So in this epigenetic landscape concept, the cellular developmental process is viewed as a ball rolling along some potential surface. Each point here represents one gene expression pattern. Of course, how the ball roll will depend on the shape of the landscape. And the shape of landscape is actually determined by the gene interactions or the gene regulatory networks. So this red picture is give an illustration. On the top, you have this landscape. On the bottom, you have this complex gene regulations which determine the landscape. But this is only an illustration. So what is this? What is really this landscape about? So in physics, we know there's a concept for the energy function or the potential. Suppose we have this from a previous network, we have this model. Here is a ODE model, for example. The X represents some concentration or concentration. F is the driving force determined by the gene regulation of the gene networks. So of course, F can have different form. I think many researchers have been working on this field to construct the mathematical model. But here, for simplicity, we just denote it with F. So for the ODE model, a traditional way to analyze it is to use this linear stability analysis around the fixed point. But we know for the linear stability analysis, we give the local information. But here, what we really want to study is the transition between the fixed point or between the basins, because you want to study the circuit transition. But this problem actually needs a global characterization. So that's why we want to use this landscape or energy function approach to study it. Because if you have this energy function, in principle, you have the global property of the system. Actually, this concept is not new. For example, in 1930s, Wright colleagues studied this evolution with this fitness landscape concept, and in 1980s, Hopfield proposed the Hopfield neural network. Also, he proposed the concept of energy function. Also, in 1980 to 90s, the energy landscape approach has been applied to study printed folding, which has been very successful. But here, what we want to study is the biological or chemical networks. Take the printed folding as an example. There's a difference between the printed folding and the chemical networks. For example, for the printed folding problem, the driving force F can be written as some potential gradient. But for a chemical or biological network, usually we don't have this form. That means F cannot be written as some potential gradient. Then can we still use this landscape approach to study this problem? The answer is yes. In 2008, the team Yang and Sudipta group, their group proposed some approach for the landscape and the flux decomposition. And later on, we extended this approach to a high-dimensional system in our 2014 paper. So I don't have time for the detail, but basically, for the gene network system, which is a non-equilibrium system, the stochastic dynamics of the system can be decomposed to two terms. One is from this potential gradient, another is from this probability flux. Okay, we have this decomposition theory, but still people need to want to quantify this landscape. So different researchers have been working on this field to construct landscape from the model. Here I list some of them. We have also been working on this problem. So I will just briefly introduce our approach. We start from the biological data, which basically we will have this structure of the gene network. Then we construct the model. Previously I have shown that we can have this OD model. Of course, to consider the effect of noise, we can have this stochastic model. So here, for example, F can be described by two function forms. Because we want to study the stochastic behavioral system, so we focus on this probability evolution. For example, we use the diffusion equation or master equation. Okay, there's one thing I want to stress. To construct the model, usually we need the network structure. We can get the network structure by literature mining, by searching for the experimental evidence. That's one way. Another way is the network inference based on the quantity of data, for example, gene extraction data. So from my point of view, I think the network reconstruction is still a challenging problem. Of course, there are some different approaches for this network inference problem. We have also been working on this problem. For example, we use deep learning to infer the network structure. Okay, then we have this stochastic model, and we in our 2013, 14, and 2019 paper, we have been developed the landscape construction approach. Basically, we have the stochastic Lagrange equation, which corresponds to diffusion equation, and we use some moment equation approximation to get the probability distribution of the system. And the potential landscape U is defined as minus log PSS. Here PSS represents the steady state distribution. Okay, I didn't go to the detail, but anyway, we have this general way to quantify the landscape for high high dimensional system. Then we did some application for this approach. One example is about this cell cycle problem. So the model is based on 2009 PNAS paper by Guttal. This model includes 44 variables, which is an OD model. But we change it to a stochastic model, and we get the landscape of the cell cycle. So basically, for the cell cycle, from the landscape view, the cell cycle can be viewed as a ball rolling along this ring valley shape. This shape guarantees the stability of the cell cycle. So this result also consistent with our previous decomposition theory. There's two falls. The green arrow represents the potential gradient, which drive the ball to the bottom, to the ring circle, to the ring valley. And another force is the probability flux, which is denoted by the purple arrow. The reason is simple. If you only have this potential gradient link, the ball will go to the minimum potential. It won't roll along the circle, right? So we need another force. Actually, the force coming from the probability flux will drive the ball to roll along this to boost the cell cycle. Okay, so we also provide a quantitative explanation for the mechanism of cell cycle checkpoint. Okay, this work is from previous work, so I don't want to talk more. Okay, but for this cell cycle landscape, I was not very satisfied with some point. For example, as I told you, this landscape actually is a 44 dimension, is from a 44 dimensional system. So at that time, we use a very simple dimension reduction approach, which is that we just pick two important marker gene for cell cycle and do the dimension reduction, but that's not perfect. And then in last year, we developed some approach for this dimension reduction of landscape. So because we want to have a better dimension reduction approach for the edge landscape. So for the dimension reduction, we know PCA is a common way to do the dimension reduction, but there is a big difference because we know PCA is a data-driven approach, but here we don't have any data. What we have is a stochastic model, dynamic model, and we have this distribution of the system. For example, we can have this Gaussian form distribution. So, but we can study the idea of the PCA. So the question is how can we calculate the covariance matrix based on a distribution rather than based on data? So the answer is yes. So it's actually very simple. My student just did some calculation. So the conclusion is we can calculate the covariance matrix based on distribution. And then for random variable X with multi-stability, we can calculate the mean and the covariance matrix. Then we can learn the idea from the PCA. We can, with this sigma, with this covariance matrix, we can do the eigen decomposition, and then we can do the dimension reduction. Okay, to test the effectiveness of this dimension reduction approach, we did some tests on different systems. The first example is this simple two-dimensional system. We can do the dimension reduction to one-dimensional system, and we can compare the two-dimensional and one-dimensional system. And here, the red point represents the stable states. The green point represents saddle point. We can see the dimension reduction preserve the stable points and the saddle points well. Also, we can study the quantitative behavior of landscape change as a parameter change. Here, as the parameter change, we can see both the landscape, the two-dimensional landscape and the one-dimensional landscape change, and we can define the barrier height, which quantifies the transition difficulty from one attractor to another attractor. We can calculate the barrier height at the parameter change. So here we show the comparison. The blue line represents the barrier height in one-dimensional landscape, and the red line represents barrier height in the two-dimensional landscape. You can see they are very consistent. This indicates that the dimension reduction preserve the stability property well. We further tested in some more complex system. For example, this system is based on this 2014 science paper for the development, and we get the landscape. We do the dimension reduction. So this system is a bistable system, and then we also want to do the comparison between the original system and the two-dimensional system. So for this two-dimensional system, after dimension reduction, we can calculate the barrier height. For the original system, I have to emphasize there's one issue, which is sometimes it's not very easy to calculate the saddle point and barrier height for high-dimensional system. So in this case, we just turn to calculate the transition action, which has a similar critical meaning with the barrier height. The transition action can be numerically calculated based on this formula. Okay, then here we show the barrier height change with parameter and the transition action change with parameter for the original high-dimensional system. we see their trend are very similar. So we conclude that this dimensional reduction approach can preserve the stability property well. There are also another thing. Here, from the eigen vector, we can see what does the PC1 represent. So for this example, we found PC1 is positively correlated with some stem cell marker. So here PC1 has a good biological meaning, which can represent the degree of stem cell state. We can also okay, we further test it in another more complex system, which is the 52-dimensional system, which is also for development. So we get the similar conclusion with the landscape after dimensional reduction, and the original system has a similar trend for the pressure height. Also, if we explore the PC1 and the PC2, we found PC1 is actually represent the stem cell marker gene, and the PC2 actually correspond to the differentiation marker gene. So there's one thing in the data-driven PCA approach, which is sometimes we will have difficulty to explain the physical or biological meaning of this PC1 or PC2. But here, from our model-based dimensional reduction approach, we see this PC1 and the PC2 has some good meaning, have some good biological meaning. So we believe that this approach may provide some general way to study the dynamical system, specifically for the gene network. Okay, so the next thing I want to talk about is related with the cancer. We know the cancer has a lot of hallmarks. One important hallmark of cancer is related with metastasis. And recently, there's many work on this EMT or epithelial-mesenchymal transition, because people think the EMT is related with cancer metastasis. And there are different modeling work on this EMT to study the transition property between E and IM state. Mohit, I think Mohit also did some very important work on this problem. Okay, there's from this previous work, there's one important thing, which is there is an intermediate state. There exists the intermediate state between E and M. So it's not just the E state and M state. There's some partial or intermediate state. So we did some work to study the relationship between EMT and the metastasis. In our 2018 paper, we brought this EMT-metastasis network and get the landscape for EMT-metastasis. We get this 4 attractor landscape for this system. So the major conclusion is we found the metastasis process is a staged process. So basically, the cell need to first finish EMT and then go to the metastasis. So the stage process for metastasis progression increase the plasticity of cell decision, and also provides a possible source for the tumor heterogeneity. In this work, we also propose some landscape control approach that we can use to control the landscape. So basically, with this approach, we can identify the combination of targets to control the cell fate. Okay, so we want to further ask some questions. The first question is, can we study the cell fate transition of EMT based on a more complete network because as I showed, usually people will work on the simple network to study the EMT. Another question is what is the role of intermediate states in EMT surface decision? The third question is related with this critical transition or early warning signals. I read one paper by Mohit and colleagues published in 2019 PNAS. So in this work, they propose there exists some early warning signals in EMT based on this simplified gene networks. So inspired by this work, we want to study what could be the mechanism of this critical transition in EMT. Especially, we want to study it from a more complete gene network perspective. Okay, to address these questions, we constructed this more complete gene network for EMT. So this network includes 16 nodes and 53 links. The network includes some transcription factor and also some microRNA. We construct this network based on some previous work. For example, this 2017 paper. And with this network, we can construct the OD model. For example, we can use this Q function form to construct a general model. Then we explore the parameter space for this model to search for the multi-stability, because as I said, there will be intermediate state between E and N. So for example, in certain parameter region, we can find this landscape of EMT with 6 stable states coexisting. You have E state, M state, you have IE and IM state. Here, IE represents the intermediate state close to E state, and IM represents the intermediate state which is close to M state. So we can have multiple intermediate states. We also calculate the transition path from E to M and from the M to E. We found that this transition path is not reversible. Also, we can make perturbation to the parameter to see how the state, how the stability change. So, for example, we make perturbation to parameters. You can still see this distribution of stable states. We have E, M state, and we have multiple intermediate states. Okay, the next thing is we can calculate the transition path for EMT from E to M state, and we want to see whether our transition path is supported by the experimental data. And then we get some experimental data for this transition path for comparison. Here we show some example for different genes. Here the red line represents the transition path from our model, and the green and the blue line represent the transition path from the experiments. So we made this comparison. We think to some extent our modeling results are consistent with experiments. Since if you compare these 2 experiments themselves, you found the experiment 1 and the experiment 2. They also have some differences in this transition path. So we think the transition path from our basically agree with the experiments. Okay, next we want to ask what are the possible role of intermediate states. We actually found in certain conditions, in certain parameter regions, the intermediate state can accelerate the EMT. Here, the way we study this problem is we calculate the transition action. We can calculate the transition action from E to M. here the transition action is related with the transition probability. A smaller transition action means a larger transition probability. So if we change the parameters, we will see the system will display different number of stable states or different number of intermediate states, and we sample the parameters, we can get the distribution for this transition action for different cases. Here is the result. The x-axis is the number of intermediate states. The y-axis is the transition action. So we can see as the number of intermediate states increase, the transition action will first decrease and then increase. That means it seems there is a optimal transition action at optimal transition action as we change the there's an optimal number of intermediate states for the least transition action, which means in this case, if we have two intermediate states, it will correspond to the least transition action or the largest transition probability. Okay, that means the two, in this case, the two intermediate states will accelerate the EMT. Of course, I need to emphasize this: number two is not something special. Depending on different parameter region, you could have different number of intermediate states as the optimal number for the least transition action. But this the point is that we can see that in certain number of intermediate states, it will accelerate the EMT. Actually, we found that similar conclusion has been drawn based from some other work. For example, this 2020 paper, but they didn't work on the gene network. They get this conclusion with a simplified state transition model. So our gene network model provide further evidence for the intermediate states accelerating EMT. For this conclusion, I think there are two factors that need to be considered. The first thing is the currents of intermediate states increase the plasticity of self-heat transition, and the stepwise transition will lower the total transition action from E to M. And another factor is because there's another outcome for the increase of number of intermediate states, which is the system will need to cross more barriers to finish the transition from E to M. So the final outcome will be the combination of these two factors. Okay, the next thing is related with the early warning signal. As I told you, I was inspired by the previous work on this early warning signal in EMT. Here we did some exploration from our EMT model. For example, we take the SAP2 as an example. We show the trajectory of SAP2 as we increase the EMT signal. Here as we increase the EMT signal, we can have the trajectory can be divided to 4 different stages, which is E to IE, IE to AM, AM to M, and M stage. Okay, so the total process can be divided to the 4 stages. And following the traditional way for the early warning signal, we calculate some common measures. For example, the correlation variable. We also calculate the coefficient of variance and the final factor. From our perspective, I think a good measure of early warning signal from one for all these different states. But what we found is that among these measures, now there's no one single measure that can perform all well for this 4th stage. For example, if you look at the correlation, here is not good. if you look at the variance here, it's not good. But in summary, we found the final factor seems is a better indicator for the both EMT and the MET. I think we think that the reason might be these measures are mostly local measures. So can we have some global measures which might be more accurate as an early warning signal? So based on the landscape, we can calculate the barrier height between stable states. We think barrier height may provide a global measure for this early warning signal. Our expectation is that the barrier height will decrease for each stage as the EMT signal increase. Because if you have when barrier height decrease, you will have more chance to make the transition. Right? If the barrier height decrease, the state transition will more likely happen. So we get this. We calculate the barrier height for both EMT and MET at different stages. Here, the blue line is the barrier height results. So as expected, we found in different stages the barrier height decrease as signal increase. We also calculate the final factor. For the EMT process, the final factor seems to also work well because you can see the final factor increase at different stage. But for MET, we see the final factor is not always performing well because, for example, here the final factor does not increase. But for the barrier height, you can always see that the barrier height decrease. Right? You can see at different stage the barrier height all decrease as the signal increase. So we think the barrier height might provide a global and a more accurate measure for the early warning signal. Of course, I need to emphasize because here we have a dynamic model in hand. So in reality, mostly you don't have this model, but you just have data. So to quantify the landscape, we need to resort to some data-driven landscape approach. But there indeed are some approaches to construct the landscape based on the data. Okay. Of course, we can also do global sensitivity analysis to discover the critical genes in this transition process. Okay, I will make a summary. For the non-equilibrium gene network system, the driving force of system can be composed to potential landscape and the probability flux. Both landscape and the flux are important to determine the transition dynamics of the system. We develop a model-based dimension reduction approach to get the landscape. For the EMT network, we found from the calculation of transition action, we found the intermediate state can accelerate EMT. Also, our results show that the barrier height based on the landscape can provide a global and a more accurate measure for the early warning signal to predict critical transition in EMT. Okay, I'd like to thank my collaborators and my students. The dimension reduction work is done by a PhD student, Xin, and the EMT work is done by Waddington. Okay, thank you very much.
Thanks very much, Chunhe, for the excellent talk. Any questions?
Hello? Can you hear me?
Yes, Dutta, please go ahead.
Yes.
Yeah, so in the dimension reduction, actually, you have used this principal component analysis. And in that analysis, actually, what are the variables that you have considered for constructing that covariance matrix?
Do you mean how do we
calculate it is purely technical thing. How you have constructed, means what are the variables that you have considered for this covariance matrix?
Yeah, it's just you start with some stochastic variables, and we just okay. This is just some calculation. You have different stable states, and treated as a random variable. You can from the probability theory you can calculate this mean, the mean and the variance. Here is the result for the mean and the variance. So, but the detailed.
Now I'm asking what are the x?
What is the x?
What is x? What are the x?
X is just a random variable. For example, it's a gene expression, but it can be a multi-dimensional. I mean, it can be a vector representing the gene expression.
Those are the gene expression. Okay.
Yeah. Gene expression level. Yeah.
Okay. So another thing, actually, you have calculated the potential barrier.
Yeah.
Potential in that calculation actually means how you have chosen that path. Means you have calculated that path, or it is actually you have arbitrarily chosen that path.
Sorry, I didn't quite follow you.
I'm asking you that the pathway that you have shown, no, that for many cases that those paths you have calculated, or those paths are just arbitrary paths based on the potential diagram you have drawn.
The landscape is here. It's just calculated from our previous approach, and then we can calculate the barrier height because you have the local minimum and the set point. I'm not sure. Is this what you're asking?
No. So can you show me any of the path that you have shown? Path.
Part?
Yeah.
What part?
The one of the So for example here. So in the potential energy landscape, actually you have shown one path. That the path is actually an arbitrary path, or it is something you have got from some optimization problem.
Oh, you're talking about the transition path, right?
Yeah.
Transition path. Yeah. Okay. The transition path here is we have this attractor. So transition path, it means it means we can calculate the path from, for example, from this ESC state to the DC state. So the process is calculated based on this formula. This formula is from the large deviation theory in mathematics. Basically, you can define a transition action which minimizes this distance.
Okay, so ultimately it boils down. Yeah, ultimately it boils down to that optimization problem. Yes.
Yeah. It became an optimization problem. We use some numeric way. Yeah, we calculate based on this reference.
Okay, thank you.
Thank you.
Are there any other questions? Chunhe, can you please explain the argument that you had about the intermediate states accelerating EMT? You had a graph where you had number of intermediate states on the x-axis.
Here?
I think later you had the number of intermediate states on the x-axis in that graph. We were talking about action. Yeah.
This one?
I think one or two slides later.
Here?
This one, yeah. Panel F, can you please explain that?
Yeah. So we can for this system we can sample the parameter, right? We can sample the parameter. So for different parameter, you can have like six landscape with 6 stable states. As I showed before, you can also have 5 states or 3. So we can do the statistics, and then we can get and for each parameter we can calculate the transition action from E to M, because the problem we want to study is what is the role of EMT of the intermediate state in EMT. Okay, so we just do the statistics and classify this different parameter. For example, if some parameter has 0 intermediate state, we just put it here, and if it has one intermediate we put it here, and then we have this distribution of the transaction. So this F picture is a summary of the first 5 picture, and it shows the average of the transaction as well as the variance. So F is a summary of previous 5 picture. So from F, you can see at a different number of intermediate, you have different transaction. Okay, so there's a minimum transaction when the number of intermediate is 2, because small transaction means higher probability. So basically here it means if you have 2 intermediate states, it will have the largest probability from E to M. Then we conclude that intermediate state can accelerate EMT. But you also I will also emphasize this depends on the situation, because if you have 3 intermediate states, it does not compare with the 0 intermediate state, it does not accelerate EMT. So this number, I mean, what is the optimal number of intermediate states for accelerating EMT will depend on the specific situation. Did I explain clear?
Yeah, sure. Thank you, Chunhe.
Thank you.
Are there any other questions? Okay, if not, let's thank Chunhe for an excellent talk.
Thank you very much.
Thank you so much, Chunhe.
Okay. Let's stop sharing.